Okay, well, welcome everybody to today's mini-symposium on stress and tumor recurrence. We're going to have two speakers today, and I'll introduce them in a moment. Each one is going to present for 30 minutes, and then we'll have an additional 30 minutes at the end for questions and discussion. So please hold your questions until after the speakers have finished. And during our discussion, Ziba will be on hand to answer questions about the OMSC core, and we may have other core representatives on hand as well to talk about some of the requirements of this kind of research and what we have available to us here. We're going to wrap up by 1:30. So this mini symposium is a new format we're trying, and our goal to bring together basic and population science researchers for collaboration. And we're focusing on topics where there's strong potential for innovative multidisciplinary research. There are two additional mini symposia coming up in this series, one from noon to 1:30 on April 19th, and that one is with Anna Gomes, who will focus on cancer and aging, and one from noon to 1:30 on June 7th with Julie Kim and Melissa Simon, and their presentation will focus on health disparities in gynecologic cancers. You should plan to attend both of those mini symposia if you're interested in responding to an upcoming Lurie Cancer Center RFA that will provide funding for basic science and population science team research. Attendance at all three mini symposia will be required for those applications. And if you have any questions on that, please reach out to me or to Jody. I think Jody is the one with more answers to that. But you'll be getting more information about that. So we're very excited to welcome our two speakers today. First, you're going to hear from Dr. Dmitry Gabrilovich, who's Chief Scientist in Cancer Immunology at AstraZeneca. So Dr. Gabrilovich has held a number of leadership positions in his field, and he's recognized as a highly cited researcher. He's also a very prolific researcher with over 250 peer-reviewed publications. And in 2020, he was awarded a research professorship from the American Cancer Society. This is a lifelong designation, and it's the most prestigious award that ACS makes, in that it recognizes leaders in innovation and influential fields that are changing the direction of cancer research. Dr. Gabrilovich conducts research on methods that tumors use to suppress the immune system and on the development of new effective methods for using immune therapy in cancer. Today, he'll present his recent work on regulation of tumor dormancy by stress-activated neutrophils. After Dr. Gabrilovich's talk, Dr. Paige Green will present on neuroregulation of cancer, implications for cancer survivorship research. She is Chief of Basic Behavioral and Psychological Sciences branch in the Behavioral Research Program, Division of Cancer Control and Population Sciences at the National Cancer Institute. Dr. Green's NCI career has focused on cultivating research on behavioral, biobehavioral, and psychological science mechanisms and elucidating their translational relevance for cancer prevention and control. So really our perfect speaker for today to follow up on Dr. Gabrilovich's talk. Her research program is multidisciplinary, spanning preclinical animal models and clinical studies of biobehavioral mechanisms and interventions. She's also the current president of the American Psychosomatic Society, in addition to other leadership positions and awards in her field. So I'm gonna right now turn the virtual stage over to Dr. Gabrilovich for his talk.
Okay, I hope things are going well. So good afternoon, everybody. I'm very happy to be here. Thank you, Christine and Jody, for inviting me. One word of introduction that although I am an employee of AstraZeneca, what I'm going to discuss with you today does not concern any of AstraZeneca products or research activity actively pursued at this moment. So, okay, that's me. So what am I going to do? Well, my research in academic world and now in industry, in the research lab, I'm pursuing was always focused on myeloid cells. And obviously, this is absolutely fascinating area of research, both basic and preclinical, because basic and clinical, because this is the most abundant cells in our body outside probably red cells. And they control multiple different mechanisms of regulation of immune responses and our protection against bacteria and viruses and other pathogens. So, and they have a fascinating biology. What I'm going to share with you in next 20-25 minutes is one story, the story which we've been working on for about 3 years and continue at this moment in a different angle. And the story has something to do with the fundamental problem in clinical oncology, specifically in very many conditions when a patient successfully treated with surgery in early stages or chemotherapy and radiation and have complete disappearance of primary tumor. Sometimes in the clinical designation is called NAD, it's no evidence of disease. But 3-4 years after that, the patient recur, the tumor come back, sometimes in a distant site, and treatment of this condition become pretty difficult, although there's some inroads there, but it's a very difficult situation. So this slide is illustrate this point. And the important idea behind it is that, and the data behind it is that sequencing demonstrated that the genetic makeup of the tumors showed up 2-3 years, 4 years after primary was successfully treated, or how we think successfully, was identical to the primary tumor. So the mechanism by which that happens has been well understood. At least we believe that it's well understood. It's a mechanism called tumor cell dormancy, and the situation when the tumor metastasized very quickly, it's early in the tumor development, and then seeded different organs, or different organs, obviously, it's provide for very hostile environment for tumor, don't allow tumor cells to grow, they're sitting there, not proliferating, and that process has a very clear molecular events, which I very briefly outlined in a couple of slides. What is not clear is why the tumor cells start proliferating again. And that's what we try to figure out. Tumor cell dormancy is can be subdivided into 2 major kind of pathways. One is the quiescence and another one is senescence. Quiescence is the reversible state of growth arrest of the tumor cells, and the senescence is considered in the classical definition as irreversible, usually associated with age-related events and how we call it replicative senescence. And that's it's irreversible because it's the reason because it's called irreversible because the shortening of telomere. And because of shortened telomeres, the cells do not have a capacity to replicate. In recent years, it's become clear that that definition required some correction because there is an early stage of senescence or senescence-like conditions where the tumor can be reactivated to proliferate even when they have a classical definition of senescence. So this slide I picked up from a couple of different reviews, just to give you very basic outline of molecular differences between quiescence and senescence. They are quite clear, but interestingly enough, this arrest and cell cycle is not that clear distinction between quiescence and senescence, and initially thought that we can't differentiate, but it turned out that senescent cells can be arrested in different phase of cell cycle, and G0, of course, it will be the major one, senescence characteristics, and one of the big differences is that senescence is driven by genetic events, oncogenes or affecting suppressive gene, tumor suppressive gene. Quiescence is more like a growth factor regulated process. So, and this slide just to give you a very brief outline of some DNA damage response marks, epigenetic marks, which people use for characterization of, for instance, senescence phenotype. And the reason I just show you it because we use those data to identify the mechanism of the process we're dealing with. So the myeloid cells, as I already said, is a critical element of our protection. And these myeloid cells respond to different pathogens in form of toll-like receptor ligand or pathogen or damage associated partner recognition receptor. This leads to the mobilization of neutrophils and monocytes as the main representative of our myeloid cells, going to periphery where they get activated and protect us from bacteria infection. So that's a very well classical mechanism which protects us as a species for tens of thousands of years, right? No, millenniums actually. But in cancer, the situation is quite different. And the myeloid cells is developed in response to the cytokine growth factors, constant stimulation leading to very consistent but modest expansion of myeloid cells. And as a result, these myeloid cells developed in a slightly different fashion. Specifically, they are less aggressive in production of in phagocytosis, production of antibacterial agents. However, they are very potent in suppression of immune responses, and they produce different metabolite, which usually normal neutrophils and monocytes produce very little, like prostaglandin 2, arginase, and so on. And as a result, these cells become immunosuppressive. And that's what about 12 years ago, the term was introduced, or 14 years now, called myeloid-derived suppressor cells, the cells which suppress immune response. And it's now studied very aggressively in cancer and infectious diseases, and COVID-19 in one of the big example, and many others. So it's a big area of research, but we are not going to talk about that in details. The reason I brought it up because it's kind of provide us with a tool to study the mechanism we are interested in. And the final summary slide for you is just to show you that in any given moment in patients, since we talk about cancer, in cancer patient in particular, classical neutrophils, the ones which protect us from pathogens and are usually not tumor promoting cells, so in some instances could be anti-tumor, they coexist with pathologically activated immunosuppressive PM and MDSC, the cells which has a different transcriptome and proteome and different function. It's a stable state of pathological activation. And the balance between these cells is the same as the on monocytic side. It's between monocytes and monocytic MDSCs. Is basically shown to be very critical for the clinical outcome and response to the therapy. So come back to the general idea. So as I mentioned to you and try to show you that quiescence and senescence, and primarily quiescence, has been heavily implicated in that dormancy, tumor cell dormancy, which allow the cells to sit for a long time. Question was why they get activated. And that's actually is not trivial because common usual suspect like infection, inflammation, wasn't confirmed epidemiologically to be the mechanism which associate with the tumor cell recurrence. So in order to start this process, I asked my former postdoc, Michela Pirega, about 3 years ago, challenged her with this, and we developed the strategy. So Michela now stay at Wistar as research assistant professor in a different lab. And she was the main driving force behind this project. I connected with David Felser, at that time assistant professor, now associate at the University of Pennsylvania. And while he was a postdoc at Tyler Jack Lab, he developed this model of inducible p53 mouse system, where in condition with the KRAS and p53 deletion usually result in, not usually, it's always result in tumor growth. And in this case, it's a lung cancer. However, when you return expression p53 using the log-stop-logs cassette, p53 is upregulated and tumor progression is halted, decreased. And we thought that we can use this system to generate arrested cells. It turned out to be not that easy proposition because cells are not really easily arrested in that situation. So Michela had to utilize several tricks, and one of the biggest one was to sort early arrested cells based on cell trace violet. And as a result, she managed to keep cells non-proliferating state for at least a week in culture. And you can see this illustration of this process. And moreover, when she put the cells into the NSG mice, the tumor, because these cells have luciferase reporters, so we can trace them, they do not generate tumors, or at least visible lesions, for at least a month. And proliferating cells produce lesions very quickly within at least within 2 weeks. So we ask what type of tumor cell dormancy this p53 inducible cell arrest has, and all data pointed out on senescence-like phenotype, expression of beta-gal, expression of some epigenetic marks like trimethylated histones expression, and interestingly, cyclin A and laminin B downregulation. And interesting is in vivo studies, you can see that the tumor cells are still sitting in the lung, but they don't proliferate. So this is arrested cells in this situation of these mice. So we cannot see them as a lesion, but we can see single molecules because they also express GFP. So in the tissues, you can see that. So that was one model, which I will show you in a second, but this another 3 other models is chemotherapy-induced senescence. It's a little bit more clearly associated with the senescence-like phenotype. We managed to generate these models or kind of adopt this model by treating Lewis lung carcinoma. It's a mouse, ovarian cancer, and lung cancer in humans by using cisplatin. As a chemotherapy, you get them to the growth arrest. As you can see, they're arrested in G2M, G0, G1's phase, and you can see that we have a very clear beta-gal as a clarification or manifestation of senescence-like of phenotype. And the other marks which associated usually with the senescence type of cells. So we now know what we're dealing with, and we have models, and that's what we can start looking at, what exactly reactivate this tumor cells. So first, we took different type of myeloid cells and lymphoid cells from tumor-bearing mice and asked which one can do it, can do the job. And it turned out that the only cells which really can do the job was polymorphonuclear MDSCs. So these are pathologically activated neutrophils. You can see here that they cause reactivation of proliferation of tumor cells, but not neutrophils from healthy mice. And we can recapitulate this in vivo by treated mice with adoptive transfer of these cells. And they really become proliferating cells and lose the all marks of senescence. That's great and nice, with one big caveat is that normal in tumor-free mice or in tumor-free healthy individuals after, well, NAD, people without evidence of disease, there is no PMN-MDSC present. PMN-MDSC is a pathological state of activation of neutrophil associated exclusively with presence of cancer. So there was something changed in normal neutrophils that drive this process. And what we found was, one of the biggest hallmark of PMN-MDSCs, which discriminate them from neutrophils, is dramatic upregulation of S100A89. It's pro-inflammatory proteins, which are abundantly present in neutrophils, but dramatically increase in PMN-MDSCs. And importantly, this is basically part of any signature, genomic signature of PMN-MDSCs. So we asked the question, can we reproduce this phenomenon if we take neutrophil from naive mice, healthy mice, and add S100A9 to them? And lo and behold, we could completely recapitulate reactivation of proliferation. And it happens only if we add S100A9, not IL-6, IL-1 beta, TNF alpha, or LPS. And when we do the same experiment in vivo, in S100A9 knockout mice, So when we take PMN-MDAC without S100A9, we lose the effect in vitro and we lose the effect in vivo, because you see that in from 75% tumors, mice which develop tumor when we inject the PMN-MDACs, only 23%, which at the level of control, happened when you inject PMN-MDAC from S100A9 knockout mice. So S100A9 turned out to be critical. So the question is, why is S100A9 then released, and what causing those changes in S100A9? Because it's an extracellular fragment. And that's how the stress come along, because I was interested, we know that as stress has been involved in tumor progression. It's very old data. There is nothing really known about recurrence, but I always assumed that it could be the case. So we try by using several hormones associated with stress: epinephrine, serotonin, and cortisol. And all three induce very massive and very quick release of the protein S100A9 from neutrophils. And that release really associated with the reactivation of proliferation of dormant cells. Simply addition of norepinephrine was sufficient to reactivate dormant cells. And it was completely canceled with the beta-2 blockers, which cancel the effect of this hormone. And that phenomenon is not reproduced in knockout mice. So basically, if you take neutrophil from S100A9 knockout mice, this effect was not present. So basically, it's confirming that S100A9 is downstream of stress and hormones. And this slide show you using the human and mouse system. S100A9 in combination alone and with combination with norepinephrine completely recapitulate which I showed you before using the chemotherapy-induced senescence mechanism. So basically indicating that S100 is indeed downstream of stress hormones, and that mechanism is really valid. The problem is, scientific question is, how S100A9 really can regulate the activation of neutrophils. So what it does for neutrophils. We have several different hypotheses, net formation and potential ER stress response and so on. None of them was shown to be involved. But what S100A9 does is upregulate activity of myeloperoxidase. It's another very important constituent of neutrophils. It's a very important part of the neutrophil activity. And it turned out that S100A9 drive this activity very strongly. And moreover, that if you take myeloperoxidase knockout neutrophils, you cannot recapitulate the effect of S100A9. So we have a link: stress hormones, acute release of S100A9, in paracrine or autocrine fashion, activation of MPO. Great. So how it may work out. But first, let me show you that we validate this data in vivo, in stress, in the condition of the stress. So we used one of the common mechanism of stress induction. It's immobilization stress. It's 4 hours per day, 5 days per week, or for 3 weeks immobilization and specific apparatuses like Wixlin tube. And then we of course we first inject tumor into the mice, and that leads to the very strong activation of tumor growth. So this work was done in collaboration with Yulia Nefedova. She's associate professor at Wistar, and her postdoc Cindy Lin. They help us with some aspect of this work, in addition, of course, to the main Michaela's work, which I constantly stressed as an important factor. So basically, this is the busy slide, and I have not that much time. Therefore, I just summarize what here is. And this published data a couple of months ago, so you can verify my statement. Basically, stress by itself was sufficient to drive reactivation of tumor lesions, basically. And if you take knockout mice, both MPO or S100A9, you cannot see this phenomenon. So that's basically the main findings from these experiments. So now, I described to you this line of research from stress to the S100A9 release and to activation of myeloid peroxidase. But how it affected tumors? That was the big question. One of the biggest findings what we have, contrary to standard activation of neutrophils, which with LPS and others, which lead to degranulation of the cell and death almost immediately. Norepinephrine activation of neutrophils does not lead to the death of neutrophil. Basically, neutrophil live as long as their natural life is. So it's not a degranulation, it's just release of this S100A9 through the non-classical way of clathrin-mediated release of the protein. So a question is what happens within the cells. And what we found, we collaborate with my old collaborator and friend, Valerian Kagan, who is a lead lipidomic center in the University of Pittsburgh, and his associates, both of them research professors there, Dr. Turins. They found that that kind of treatment result in accumulation of very interesting form of plasmalogens, lysophosphate-anolamine, lyso-PE. And these lipids dramatically upregulated in the neutrophils from stressed or S100A9 treated. These molecules was sufficient to drive proliferation of dormant tumor cells. So this slide just basically illustrates this point that you can recapitulate this by extracting lipids from the activated neutrophils by stress. And that effect was completely lost if you use myeloid peroxidase knockout neutrophils. When they made these lyso-PE species in vitro by manipulating the lipids with myeloid peroxidase and then send it to us, we could manage to reproduce the effect of activated neutrophils completely. So we have now this next step of this model. So, but how lyso-PE can activate proliferation of tumor cells? And that would help from Andrew Kosenkov, who is a bioinformatician at Wistar, come along, because we did RNA-seq analysis of tumor cells which were never arrested. It's called KPR. And the tumor cells which were arrested and then reactivated, they're all proliferating pretty similarly after that. But it turned out that they still were very different. And one of the biggest difference was the upregulation of the FGF pathway, it's fibroblast growth factor. And that's manifested in upregulation of the receptor, FGF receptor. It's manifested in upregulation of actual FGF itself. The reason why it matters, because it's very well established in the lung cancer, FGF pathway is involved in cell proliferation, tumor cell proliferation, and maybe actually involved in many, more than just lung cancer, but in lung cancer, it's especially true. So when we went back and used one of the selective FGFR receptor inhibitors, we found that that inhibitor completely canceled S100A9 or stress-induced reactivation of dormant tumor cells by neutrophils. So we done this in vitro, and this results in vivo. So when we induce stress in the mice and then add inhibitor of FGF receptor, we managed to completely abrogate tumor lesion formation. So suggests that we have that kind of fundamental link between these two events. So, what we tried to do next was to see whether those things holds up in clinical situations. Of course, we did use clinical samples, as I mentioned to you, in human lung cancer and ovarian cancer, where we used human neutrophils for the experiments, but that was in experimental ex vivo. Here, what we did, I contacted Dr. Harvey Pass. He's the head of thoracic oncology at NYU, who collect samples of serum from his patients who were undergoing surgery at stage 1 or 2 patients with non-small cell lung cancer, and asked him to give us samples, which he kindly provided us with. And using help and collaboration with Thomas Vogel and Johannes Roth from University of Münster in Germany, we ran this S100A8 A9 recombinant, heterodimer S100A9 and A8 in the serum of these patients. When we've done it, we found that the patients who had a late recurrence, they have much lower expression, the level of S100A9 and A8 in their serum. And when you do the survival analysis, it's a recurrence-free survival, you can see that there was a pretty clear separation of high, which go much faster recurrence, early recurrence, and relatively low presence of these proteins associated with much longer duration of recurrence-free time. So basically, this association study indicates that this could be something clinically relevant. And we also find the statistical significant correlation between the level of S100A8 and 9 in patient serum and norepinephrine in the same patients, suggesting that this concept, which I just presented to you, was probably real. I will finish my talk with this schema, which is really what we think may happen in the recurrence. So when the patient has a dormancy, dormant tumor cells undergoing stress, which we all subjected to on a constant basis, fear of death for cancer patients considered one of the most powerful stressful signal. And then neutrophils are going in all different organs. And the norepinephrine obviously can reach them anywhere. They release S100A9, which in paracrine or autocrine's fashion bind to the receptors. I didn't mention those receptors, they're known. It's toll-like receptor 4, H. And they bind and upregulate myeloperoxidase, which causing oxidation or hydrolysis of the lipid species and produce those lyso-P, oxidized lipids lyso-P, and then they bind to the receptor, which we try to figure out which one, and causing upregulation of fibroblast growth factor, leading to MAP kinase upregulation, which is known downstream from the FGF, and driving cell proliferation. So, and this process is not like binary system, yes or no. Neutrophils come to tissue and they die within a couple of days, sometimes even earlier. So basically, if you don't have a stress signal at that time, nothing will happen. So that's what may explain that it may take years, it may take more than that to have this process happen, because you need to have a combination of things. The stress need to be present and constant, because this kind of interaction does not happen immediately. And just simple activation of the binding to the receptor of the lyso-P cannot change immediately on the dime the activity of the tumor cells. So that's may explain the slow process of accumulation of those changes in peripheral tissue. So I already acknowledged everybody who did this work. We have people who continue that now in AstraZeneca, but I think I will stop here, and thank you again for your time. And when time comes, I'm happy to answer your questions and participate in discussion.
Thank you very much, Dr. Gabrilovich. And as one of the population science people, I really appreciate that final slide, which helped me see all the pathway and was incredibly helpful. Next, I'm going to turn it over to Dr. Paige Green, who's going to speak on the neuroregulation of cancer and implications for cancer survivorship research.
Thanks, everyone. All right, let's see. Okay, can you see my slide deck in the correct way? Okay, great. And can you hear me? Okay. Oh, great. Well, thank you so much for having me. It's really a pleasure to follow that presentation. I am not a basic scientist. I'm a biobehavioral scientist, and since coming to NCI, I've really been trying to cultivate a portfolio that helps us understand the role of stress and psychological experiences and how they modulate cancer outcomes. So my goal today is to just briefly introduce you to me, because I think it may help, shed light on my perspectives, and then highlight just our development of this federal portfolio in the neuroendocrine regulation of cancer, and offer some implications for cancer survivorship. Okay, so, all right, there seems to be a delay. All right, so my slides are not advancing. Is that?
Can you try to exit PowerPoint?
There it goes. There is such a significant delay. I'm so sorry. And I am hardwired, so I'm not sure what's happening here. As you know, I'm at the National Cancer Institute. We're the leader of the National Cancer Program. We're the largest funder of cancer research, of course, in the world. And I just wanted to sort of highlight our role in this enterprise. And from my particular position, I'm in the Division of Cancer Control and Population Sciences. So let me just orient you a little bit. The mission of our institute is to really develop the knowledge base that will lessen the burden of cancer in the United States and around the world. And I contribute to that mission as a servant leader of the basic biobehavioral and psychological sciences branch. And I'm nested within the behavioral research program in the Division of Cancer Control and Population Sciences. And I know that Siobhan is on, and I saw Bonnie Spring, so they are, and Christine, are very familiar with the work that our division does. So although I'm in the behavioral research program, you will know that we have several other programs that are really instrumental in generating the knowledge to reduce risk, incidence, and death from cancer, and also to improve the quality of life of cancer survivors. So we have programs and disciplines such as cancer surveillance, epidemiology and genomics, health care delivery research, cancer survivorship, and in the behavioral and biobehavioral and psychological sciences. So let me just give you a little bit more insight into my branch in particular. And we really seek to support research that's fundamental to understanding human behaviors and social contexts, social dynamics, decision-making, and clinical practices that are associated with the cancer control continuum. And when I say that, I'm referring to cancer etiology, prevention, diagnosis, detection, treatment, survivorship, and even the end of life. And we in particular are unique in that we cultivate a portfolio of basic experimental studies that involve animals. So Christina mentioned our preclinical portfolio, and humans, also observational studies and mechanistic clinical trials. And this aspect of our identity has become exceptionally important over the last couple of years. We're really seeking to make sure that the research that we fund really engages individuals and segments of the US population that have been excluded, marginalized, underrepresented from the sort of biomedical, behavioral, clinical, and social sciences research enterprise. For us, that's particularly important because we are a branch that supports fundamental knowledge, and without having a diverse portfolio of individuals and communities that contribute to that knowledge, we run the risk of perpetuating knowledge that is unreliable, not valid, and not generalizable. And in that goal, we also seek to nurture and sustain a diverse pool of talented investigators. We have become increasingly aware of the importance of lived experience and the importance of social identity and how researchers that we fund engage in the scientific enterprise. So I just, it's become so important to me that that now I'm incorporating this into most of my talks, because I want individuals to know how dedicated we are to doing that work. So in terms of our swath of our portfolio, we really do work towards developing the knowledge base that will lessen the burden of cancer. And we do that by cultivating what we call a use-inspired psychological science portfolio. And that really is focused on the phenomena and social and psychological phenomena that you see listed on the slide. In particular, their transduction through biological pathways and how such processes may actually moderate or mediate the associations amongst other predictors, cancer risk, and cancer outcomes. And we really hope that use-inspired basic research will improve our understanding, explanation, and observation and prevention of cancer and its sequelae. So I joined. Look at me, I look so very young now. No gray hairs, but I joined the National Cancer Institute in 2001. And we were a very small branch, and my branch chief, Michael Stefanik, who has transitioned back to academia, really tasked me with exploring the mechanisms by which brain, the nervous system, and the immune systems interact and impact each other, and the relevance of those interactions for cancer prevention and control. And that was really a very interesting and challenging time, because the nervous system and the immune system, for that fact, were considered relatively inconsequential for cancer initiation, progression, and metastasis. And cancer immunotherapy in 2001, I'm exaggerating a bit, but was really thought to be on the fringe of science fiction. So I hope to really just walk you through some very high-level insights from research from our portfolio that really aimed to elucidate how chronic stress exposure regulates aspects of the cancer control continuum. And by this, I mean specifically promotion of cancer, progression of cancer, and metastasis. And I hope to end by discussing the implications for cancer survivorship disparities, particularly for those that experience a life course of stress associated with low socioeconomic status, or what we now consider the socially constructed consequences of race and ethnicity. So you all are probably well aware, in 2008, the World Health Organization named stress as one of the 10 top social determinants of health inequities. And the frequency, intensity, and duration of stress exposures are, as you know, not equally distributed across and within populations. Low-income individuals and members of race, ethnic groups are disproportionately exposed to stress across their lifespans and through many life domains. And I've just bulleted some that are here. And you'll notice that they're sort of binned by those that are more structural in nature and those that can be considered more interpersonal in nature. But these populations that have this differential exposure to stress really face greater threats to their health, safety, and economic advancement. Those of low SES and members of marginalized groups live and work in social and physical environments that increase the likelihood of exposures to threats, and also on top of that, decrease the availability of material and social resources necessary to address those threats and have positive effects on their health. So racial and ethnic discrimination compound these effects by increasing threat exposure and creating barriers to the development of needed resources. So stress occurs when individuals face threats, demands, but do not have the sufficient resources to respond to those threats. And individuals facing economic and social disadvantage experience, of course, more subjective and objective threats. I love the schema that was developed by the American Psychological Association Working Group on Stress and Health Disparities. It really outlines the relationship of persistent chronic stress to the onset and course of mental and physical outcomes. So, the schema really attempts to capture the effects of social context on individual psychological processes, including processes involved in the perception and interpretation of the self and the social world, and that's represented by social cognition, the underlying processes that govern attention, planning, memory, inhibitory control, which I call cognitive control processes, and then also how stress affects brain and physiological systems, including epigenetic and immune functioning. And lastly, the effects of stress on health behaviors. And together, these stress processes can increase the risk for new threat exposures, undermine personal and social resources, prolong the psychological and biological effects of exposures, and produce changes in physiological systems and health behaviors that impair health. And this is a complicated, somewhat slide, but I just wanted to highlight this. This is work that my postdoctoral fellow, Karina Duffy, and Kate McLaughlin published, I believe, in PNAS in 2017, in which we were trying to articulate how early life exposure may be related to addictive or the types of behaviors that we know are associated with cancer risk. And so we hypothesized that children exposed to early adversity characterized by threat or deprivation without adequate resources or access to those resources show altered patterns of brain development, neural development that increase their susceptibility to chronic diseases. And this in part increases the tendency to engage in health risk behaviors. And so we propose neural adaptations to early life adversity can affect affect, emotion, cognitive, and reward networks of one of which, of course, is stress. And so you see through the physiological changes that are crudely, represented on the slide that, smoking cigarettes, which is the #1 besides age risk factor for cancer, drinking alcohol and overeating highly palatable foods, further heightened emotional reactivity. They hinder emotion regulation, increase delayed discounting, and blunt reward responsivity. And this leads, we hypothesize, to a positive feedback loop for behaviors associated with disparities in chronic disease, morbidity, and mortality. And I would have to say that this is one of the most joyous things of being at NCI. We can publish things like this and then expect scientists and academic communities to either challenge us by their data or produce data that meet sort of our position that we propose. So that work that I just highlighted really represents one conceptualization of stress processes and potential risk for cancer and other chronic diseases through brain development, neural adaptations that read out as psychological traits that increase our susceptibility to engage in health behaviors as regulatory strategies. And we know indeed that smoking, alcohol consumption, sedentary behaviors, dietary patterns characterized by the consumption of highly palatable foods are associated with the incidence of most common adult cancers. However, I am on the other side, in which I really resonate with this quote by my friend and dear colleague, Steve Cole, who actually should have been presenting to you. He would have done a much better job than I, but I'm his pitch hitter. So here we go. And he says, with all we know about how stress aggravates cardiovascular disease, promotes viral infections, exacerbates metabolic diseases, halts reproduction, and regulates the normal function of virtually every cell in the body, why in the world would cancer cells somehow be exempt? And I maintain, and as Dmitry highlighted earlier, they are not. So albeit what I'm about to summarize really lacks specificity at the molecular level, I'll encourage you to read this paper that was published in Nature Reviews Cancer with Steve as our first author. And then we collaborated on a few other papers that you see highlighted that really attempted to put a heuristic framework out into the field about how biobehavioral at the time, as we were calling it, influences could have significant biological consequences for cancer progression and metastasis, and of course, survival. So there appear to be four, and this is going to be really crude, four pathways by which the chronic fight or flight stress biology can really influence tumor biology and perhaps mediate disparities in cancer survivorship outcomes. There's the activation of epinephrine output, allowing stress neurotransmitters to reach the tumor through blood circulation and the innervation of tissue and innervation of tumors. And that particular process has really resulted in a new field that we call now cancer neuroscience. And it's really about axonogenesis and the infiltration of peripheral nerves and fibers into the tumor. But a little bit more about that later. The other way is through the regulation of glucose and other general metabolic resources, the regulation of the innate immune system by either stimulating macrophages as an example, and the transit to the tumor and thereby promoting tumor growth and metastasis. And as Dmitry highlighted, activating perhaps tumor dormancy and perpetuating the sort of dissemination of tumor cells throughout the system. And then we also know about the regulation of the adaptive immune system. So inhibiting cytotoxic T cell responses and undermining, we believe, the effectiveness of new immunotherapies. So a lot of our proposition for this is really based on in vivo animal models. We all know, at least those in the basic scientists, that it is very difficult, if not unethical, to, over the lifespan, experimentally randomize groups to experience life stress for the significant period of time in which we can see these processes play out. So we use animal models to gain mechanistic insights. And through a growing portfolio of work, we have shown that chronic stress can accelerate the progression of breast, prostate, ovarian cancers, malignant melanomas, neuroblastomas, pancreatic carcinoma, and some hematopoietic cancers such as leukemia. So this was a schema that we developed, and I believe in 2013, myself, Susan Luckendorf. It was actually part of a special issue that I led on sort of our 10th anniversary of trying to cultivate this field. And it really just walks you through how biobehavioral risk factors such as anxiety, and I think anxiety prior to surgery is going to become substantially more important as this work progresses, how it modulates the central nervous system, leads to neural endocrine signaling, which impacts tumor biology, and thereby affects the clinical cancer course. So in the interest of time, I won't go into the specifics of how that all works, but if you have specific questions, I can attempt to answer them, okay? So what does this have to do with survivorship disparities? Well, in 2017, Lynn Bringer, Gelhardt, and Geronimus used the weathering hypothesis that Geronimus offered many years ago to guide their considerations of the factors that influence the development of hormone receptor negative breast cancers in African American women. And it really sort of highlights what I as a behavioral and population scientist know that there are multi-level influences on cancer outcomes, right? So, you know, when you look at structural factors, community factors, individual factors, and how they influence not only the type of disease that potentially is presented, but also their outcomes. So this is, I just use this to highlight, you know, one disparity that we are well aware of, that African-American women are twice as likely to be diagnosed with triple-negative breast cancer and are much more likely to die from breast cancers compared to those that they are redundant compared to. But I thought that I would use that sort of general epi picture to really just dive a little bit deeper in the time that I have into some work that we were able to support. So I want to just highlight the work that's being done by Erica Sloan. And Erica, I had the pleasure of meeting and incorporating into our NCI network on biobehavioral pathways in cancer. And the goal of that collaborative network was really to seed ideas and preliminary data that would allow this type of research, which at the time was considered fringe and not worthy of R01, you know, status, to begin to generate preliminary data that they could use to be more successful. So Dr. Sloan is a professor at Monash University and is really, I think, one of the foremost leaders in articulating sort of the biological pathways by which stress is transduced for clinical outcome. So some of her data highlights that neural signaling, you know, accelerates cancer progression by driving cancer spread. Her work from her lab really demonstrates this in the models of both solid tumors, breast and pancreatic, and in blood cancer, leukemia. And of course, as I said earlier, we use these preclinical models to reduce the intra-individual heterogeneity seen in patients and to elucidate mechanisms that we think have clinical relevance. So her work has shown that through this neural signaling pathway, it occurs by three mechanisms, and I won't go into most of them, but one of them is that there's neural signaling that increases invatopia, and then that chronic stress really remodels the lymphatic vasculature to provide routes for metastasis. Importantly, though, she also shows that neural signaling impairs adaptive anti-cancer immunity. And one implication of this is, of course, reduced effectiveness of immunotherapies. And you can see the survival differentiation between those that were exposed to immunotherapy, those exposed to immunotherapy in a stressful condition, and no treatment at all. All right, so this is a schema that was published by Anil Sood at MD Anderson and Steve Cole in 2011 that really sort of outlined their perspective on the beta adrenergic signaling pathways in cancer. I will not go into this in much detail. What I really did want to highlight by showcasing this are sort of what I consider the greatest common denominators, and how, sort of this signaling pathway leads to differences in inflammation and angiogenesis, EMT. So, the transition from epithelial to mesenchymal, impaired immune and cellular responses. Anil's work has also demonstrated the impact on anoicus and chemo resistance and resistance to apoptosis, all that we know will serve to keep that tumor growing and healthy. All right. But I highlight that also to, show early studies by Sloan and others that, beta blockade can prevent metastasis. So this is a particular study in which propranolol is used. And you can see by comparing the vehicle to propranolol on both the control and the stress rodents, the reduction in the sort of metastasis burden in the stress condition with propranolol. Right. We also know that there are many retrospective epidemiological studies that suggest that beta blocker use is associated with reduced cancer metastasis. And so similar effects are observed in humans through observational pharmacoepidemiological studies that, of course, are retrospective. This just highlights an analysis by Lee that shows that beta blocker use, like that prescribed to control hypertension, and is associated with improved cancer outcomes when patients subsequently are diagnosed with triple negative breast cancer. And specifically that metastasis to the lymph nodes and distant tissues is reduced. And we know that the number one cause after successful treatment is metastasis and its impact on cancer mortality. So we funded Erica and her group to conduct a phase 2 randomized study of a perioperative beta blocker versus placebo on gene expression in newly diagnosed breast cancer. We also funded through our pilot network the work of Shamgar Benilahu that paired a beta blocker with an NSAID and had very positive results, albeit the sample sizes are too small to have confidence in clinical significance. And although that study by Erica's group, is different than this, she has just recently published, this paper that is a combination of mouse model data, preclinical data, and also an analysis of a huge number of breast cancer patients, I believe over 4,000, and their use of a variety of beta blockers, and shows survival advantage. So I encourage you to read this paper, and it was just published and shared, I think, with me within the last week or so. Okay. So I did mention earlier that we have this new field that's called cancer neuroscience. Their focus is not necessarily totally on neuroendocrine regulation. They're more interested in sort of innervation of tumors. But I do like and appreciate this paper that was published recently in 2020. I think Paul Fernetti is on this paper. Of course, he is one of the ones that, that published, the very impressive data on adrenergic importance or signaling in prostate cancer. But he really outlines that there may potentially be several ways in which, our sort of focus on neural regulation or nerve regulation in cancer and neuroendocrine regulation of cancer may have actual patient benefit. There's the aspect of doing selective surgical denervation. There's also the aspect of using medications that are used for other purposes like propranolol in a neoadjuvant or adjuvant setting. They propose the importance of perhaps using PET scans and other advanced imaging techniques to actually image the tumor in a way to see about the associated cholinergic and adrenergic activity. And a way that I think is still puzzling to me is sort of looking at pain phenotypes and what that might contribute to outcomes as it relates to neuroendocrine regulation of cancer. I did, though, want to highlight, and although I should really be using Shamgar's work, Shamgar Benilahu is in Tel Aviv. He's really, in my mind, at least in my sphere of influence, really been the one that has very early on, I mean, I've been at NCI since 2001, so maybe as early as 10 or 15 years, was telling me, you know, you really do have to pay attention to the perioperative period. You have to understand surgery as a significant stressor that has long-term ramifications for survival. And he wasn't, of course, talking about how well we debulk a tumor, right? He was really thinking about stress as or the perioperative period as this definitive stressor that, you know, sort of causes anxiety, you know, of course, produces wounds. And you can see by this schema that was published in Nature Reviews Clinical Oncology in 2018, just the different ways that local tissue injury initiates, you know, the potential growth of residual cancer cells. It helps with the tumor cell transmission, transit to other organs, the blood and the lymph nodes, how local tissue injury can really, you know, perpetuate circulating tumor cell release and result in colonization. And then, you know, how the modulators of perioperative stress like pain, anxiety, hyperthermia, fasting may actually, you know, lead to the sort of influence on dormant and growing micrometastases. And so this is a paper that Shamgar recently published, I believe, in Cell Cancer Research. And it really sort of is, you know, it highlights really the balance that needs to be maintained between the positive and the negative from as a lay perspective effects of surgery. Surgery, of course, as we know, is the most widely used intervention, at least for initial curative intent. I believe over 60% of cancer patients undergo surgery for their initial treatment. And I won't go into this, but I think it's just a nice schema that shows how surgery may lead to the risk of metastatic disease or reduce the risk of metastatic disease. And we also know that metastatic disease is largely driven or somewhat driven by the potentiation or the perpetuation of disseminated tumor cells and circulating tumor cells. So that is a nice perspective, I think, that Shamgar offers us. All right, so I'm almost wrapping up here. And you know, I've highlighted surgery as a potential point of intervention. I think it's the one that the medical community, that the oncologists and surgeons can most aptly address. But we also do know that, you know, stress and stress exposure continues after curative intent. There is considerable data on the stress that's associated with financial toxicity from successful treatment. And so while we could have denervation and, you know, sort of, you know, the prescription as medically appropriate of propranolol and other types of you know, medications to help systemically control the neuroendocrine and neural effects of cancer, we will need, I think, a more diverse portfolio to actually deal with some of the other contributors to poor outcomes in cancer survivors. And so this schema, also developed by the APA and their work group on stress and health disparities, just outlines very crudely the different multi-level aspects of interventions that could be employed to reduce disparities in health and disparities in cancer survivorship overall. So I want to end, you know, my talk with a quote and just respect to someone that I considered very dear to me. Arthi did not at all work on this aspect of my portfolio. Arthi and I met because she was very interested in aging and our work in accelerated aging, which I wish I could present to you, but I don't have time. As many of you know, she was tragically killed in a car accident. But she has touched me and my passion for the type of work that I continue to do at NCI overall. And so when I talk about her, and I'm going to read this, I often think of a quote from my favorite children's book, which is The Little Prince. And it aptly describes Arthy's conviction as an oncologist that cared deeply about helping her patients. And I think this quote is relevant because it's relevant to those that treat survivors or cancer patients and survivors overall. And so people have forgotten this truth, the fox says, but you mustn't forget it. You become responsible for whatever you have tamed. You're responsible for your rose. And we as oncologists, as behavioral scientists, as cancer control researchers are responsible for the roses, the cancer patients, their diseases that we tame. And she felt responsible for the health and well-being of every one of her patients. And she's quoted as saying, love your patients as much as you can, give them as much as you can with all of your heart. They're in a vulnerable place, and kindness and compassion go a long way. So with that, I just want to encourage us to think of our patients as whole individuals with lived lives and social identities beyond them being a cancer patient. There are so many things that scientifically we cannot probably account for what contributes to their survival, whether or not it's exceptional survival or survival that we considered less than optimal. So I just wanted to highlight Arthy in that regard, to highlight what a great influence she's had on my wide portfolio of work at NCI. And I could not end my talk without acknowledging my partners in crime. These individuals, Steve Cole, Susan Luckendorf, and Anil Sood, and I came together to really say, what could we do to push some energy into this field, and energy that was not, being given by others. And so they were instrumental in helping NCI set up this network on biobehavioral pathways in cancer. And these are the people that we were able to fund, right? Julie Bauer and Michael Antoni, Erica Sloan, Jennifer Knight at Wisconsin, who is studying beta blocker, sort of regulation of hematopoietic stem cell transplants. And of course, a number of mice studies, because mice are essential. They allow us to sort of elucidate mechanisms, and then hopefully, as Dmitry said, also then follow up those mechanisms by doing clinical association studies. And it is only through that type of cooperation and coming together that I think we will be able to understand once and for all the relationship between stress and psychological experiences and social experiences and cancer outcomes, including survivorship. Right, so with that, I will end, except to say the work that the network did was featured, I believe, in 2019. And I'm highlighting this largely to say, wow, what a long way we've come. So this was a feature article in Cancer on the war on nerves. And I use this to just, pay ode to how far this type of science has drifted into the mainstream of cancer research. And for that, I am very pleased. So thank you.
Thank you so much. That was wonderful. And I don't want to take up too much time because I want people to be able to jump in with questions. Feel free to jump in and ask them, or you can put them in the chat and I can read them. And I can get started if nobody else has a question. The idea that this is chronic stress is really intriguing, and it makes tons of sense. And I think I think back to all the measures we have at chronic stress, and I was wondering, are there a set of measures that you would recommend as being particularly powerful and something that we as researchers should sort of keep our eyes on as we try to do something in this area?
Is that a question for me?
Yeah.
Okay. You know, no, really. I mean, I think that remains part of the sort of methodological difficulty with doing this work in clinical and human populations. We know that cancer is a disease with an exceptionally long latency period. You know, that's why, you know, I say stress exposures over the life course or the lifespan, right? So, you know, studies that attempt to, you know, only at the point where, you know, or the age group in which, you know, cancer risk is likely to be heightened, if we were to only, you know, tap into stress at that point, we likely missed a critical and sensitive time point, right?
Yeah.
So it continues to be a challenge, and really is why our network in particular tried to focus more on biological representations of stress, right? That's why we focused on neuroendocrine transmitters that we knew could be interpreted as stress mediators. But I will say that if you want someone to present to you that can really, you know, challenge you and help you in that, anyone from the Stress Measurement Network at UC San Francisco would be absolutely fabulous. They are probably the most progressive group that is really attempting to develop very rigorous methodological approaches to the assessment of stress in individuals.
Terrific. That gives us a good source of information. And I should mention that our outcomes and measurements core in the Cancer Center should be able to help with identifying measures as well.
Yes. Yes. So there is the strain, you know, that's developed by George Slavich. I believe he was at UCLA. And there have been a few papers that have been published recently using that. So I can find those resources for you and send those to you, and you can share with your group if you choose.
Thanks. And I'm going to ask another question, and that is, as researchers, we rarely are going to have a grant that's long enough to allow us to study the trajectory from no evidence of disease to recurrence. And clearly now there are some biomarkers that we should be looking at along that pathway. Now, as a social health psychologist, I have a lot of notes here of things I need to look up, but along this pathway, when we're thinking about adding biomarkers that we can perhaps get through blood samples, or I don't know, even maybe dried blood spots would be terrific. What are the ones that we should be particularly keeping our eyes on as important mechanisms?
Maybe Dmitry should answer that, because I believe part of his work actually found, was it 100 AB in blood and correlated that with outcomes or survival? Am I misrepresenting your work,
Yeah.
Dmitry?
No, but the question was about the stress, right?
Well, no, about your work, and if we want to carry this forward and do our own research on this kind of a mechanism in human subjects research, and we can identify measures of chronic stress, what are the biomarkers that you would suggest we should be looking at in research that you've studied and found in your work?
Yeah, you see, you cannot equate stress with the biomarkers which we're looking at, because they could be regulated by many other mechanisms.
Yeah.
That's why I not big. I don't follow stress itself. It's a great causing agent. It's nice, kind of first step, but in reality, what happens with the cells, tumor cells, it's really not stress itself. It's what happens with downstream. And that events, although they can be triggered by stress, they could be involved something else. So basically, as a mechanism of regulation of tumor progression and potential targeting is great in my view. However, as a biomarkers of stress is not really that great.
Oh, no.
You have very clear markers of stress, which I know that from the literature are considered reasonably good. There's test systems to measure norepinephrine, epinephrine, cortisol, all sort of things. And then when I did work with psychiatry group at UPenn, they have a lot of measurement of behavioral stress. I mean, all these pages and pages of a questionnaire and so on. They have very clear gradation of the different level of stress.
Yeah.
So I don't think the issue is the stress as a biomarker. Nothing you can do about stress in life. I don't think it's somewhere you need to investigate further, because stress will be always with us, and it could be sometimes high, sometimes low, but that's the end result of it. There is a very good clinical activity now. I'm familiar with at least one clinical trial which they use propranolol, beta blocker, to improve the effect of checkpoint inhibitors in cancer patients, claim pretty good clinical response in phase 1 trial. That's a possibility where to go with this.
Yeah.
I know that Christina, I think that Susan Luckendorf, though, has published work that's shown, VEGF levels and circulation or something is associated with survival. I think she recently had a paper. So, that's another person that I would recommend. And I'm sorry, I don't have her latest results. She just published a paper in Cancer that I haven't fully read yet. So I do think that I wouldn't call them biomarkers, though. I think, we sometimes have very different perspectives of what a biomarker is. So I agree with Dmitry in that point. I often hear from medical and, that stress is ubiquitous. Well, so is everything else. Air is ubiquitous, right? But we all have to breathe, and there are qualities to air that, that produce health risk, right? And so, while stress is ubiquitous, there are ways in which it can be managed. And, I do want to highlight that the work that I presented in no way actually, showcase compensatory mechanisms for dealing with stress exposure, and that too must be considered. So, I think, Dmitry's work nicely shows that may be one mechanism, but it is one mechanism perhaps, and there may be others that are working for or against at the same time. So, I think it's sort of being considerate of the pleiotropic sort of effects of how cancer progresses and metastasizes that gives me continued hope in this type of work.
So I appreciate that, and I am certain that I'm oversimplifying things. But let's just say I've got a big study running, and I have measures of chronic stress of various kinds that I've identified, and I would like to have biomarkers that say this person may, I want to collect blood samples and have them analyzed and say this person might be at higher risk of recurrence based on the literature. What are the things I should be looking for other than, cortisol or epinephrine or, some of the other things? And I honestly haven't studied this for 20 years. So I want to learn about this and do it, but I don't know where to go.
Well, we have that done in retrospective studies. We show that the level of S100, 8, and 9, which is the standardized commercially available kit in which measuring in plasma, can be associated, again, it's just association, can be associated with shorter recurrence-free survival in patients. So It's not exactly black and white because S100A9 release may happen also for different other reasons. For instance, if you have infection. But as I noticed, I mean, I said in my presentation that that release does not lead to the consequences we're talking about because neutrophils will be dead very quickly because that's how they function. They need to kill bacteria, they die and together happily. Stress doing this in different ways. So I would say that this could be a very good initial step. VEGF, if it's really truly associated with stress, it's another very nice marker because I work on VEGF for quite some time. And it's indeed deeply involved in tumor progression. So
Right.
it's a
Yes.
reasonable marker. I think it's much more studies are needed to figure out the consequences of stress because a lot of things was focused on behavioral and because we know what stress is. There's so much effort on therapy. And actually, I, with my primitive view on that, read a couple of papers, review papers showing that verbal treatment or behavioral treatment, how is it called in psychology, have some positive impact on patients with cancer, clinical impact.
Yeah.
So it's definitely not just a beta blocker, just many things can be used to alleviate at least partially this phenomenon. Because of my training, because of my focus, I'm looking at very primitive kind of protein level effect. So we need to find how cells are reactivated. Why not to target that particular molecule without going up? However, this intervention could be quite useful without going to the medication, right? You can do it by therapy, and that could be it could be quite useful.
Thanks.
So it's much more than just simple drug targeting.
So I have a question
But
from
Christine, can I then just say, I mean, I think Steve's work, though, you know, is important, Steve Cole, in that, you know, while he's not necessarily looking for biomarkers, he's looking for, you know, sort of divergence of gene expression and gene regulation. And I think that probably is, and relating it to actual, right, cancer relevant biological processes. And I think that's probably more useful than looking for a biomarker necessarily. So that's just my perspective.
Okay.
Yeah.
I do have, I think, one last question from Anne-Marie Flores, who is one of our audience members. And she said, for the propranolol studies, were these patients who did not have a history of hypertension because it's highly prevalent among elderly people?
Yeah, I don't know which study in particular she is referring to. And I will go on record by saying that, you know, propranolol is not the end all or be all, right? I mean, there are medical indications for the use of propranolol for some. So I don't know the answer to her particular question. So if she wants to, if we can exchange an email, then maybe I can give her some more detail about the particular study. So sorry, Anna. Or Anne, rather.
And I'm so sorry, we don't have more time for questions or for talking, but you've given us a lot to think about and a lot to read up on. And we really appreciate you joining us for these talks today. Thank you so much.
Very welcome.
Thank you.
Thanks for having us.
Thank you.
Take care.
Bye-bye, you too.